All right, so this next video is probably going to be the most complicated part of this chapter, so you might have to watch this one a couple times. But it is about bacteria reproduction. All right, bacteria can reproduce in one of two ways. We got asexual reproduction, which you can see here would be your binary fission, which we'll talk about. And you have your sexual reproduction. So let's start with asexual reproduction. Again, that's going to be called binary fission. It only involves one parent, so you only need one, like all other types of asexual reproduction. The offspring are going to be identical to the parent and each other, so it is, you can think of it kind of like cloning, but they are going to be identical genetically. Advantages: it's simple and only one parent is required, so you only need one bacteria for this to happen. And it is a preferred way for a lot of bacteria to do because it is that simple. There's no maturation period involved for it, so once the bacteria goes through binary fission, you don't just get like a baby bacteria and have it to grow up. You get an exact copy of wherever the original parent was from. Another benefit is that this is fast, especially in ideal conditions. Bacteria can go through binary fission once every 20 minutes if it is good conditions. All right, some disadvantages of asexual reproduction. There is no genetic variety. So if a bacteria does go through this and a change in the environment happens, if one bacteria is susceptible to it, all the ones that are clones like it will also be susceptible to it. For example, whenever you use Lysol to kill bacteria, it's not going to kill every bacteria. I mean, it kills 99.9%, but that's not all. So the few that are still going to be alive from it will reproduce with no genetic variety and still be resistant to it. All those that are, if everybody is, then they're all going to die right away. So unfavorable conditions in the environment can wipe out the whole population if they're all exactly the same. Asexual reproduction is binary fission. So here's our little graphic that will help you go through it. Basically, we have our bacteria up here, and we have its genetic material inside. When conditions are favorable, and it's deemed so necessary that it needs to reproduce, it's going to start to unzip that genetic material and copy it. Eventually, you're going to get two copies of the whole thing. It is then going to separate into two sides of your bacteria cell and pinch off in the middle around that cell wall. And remember, that cell membrane is very permeable, so it'll let things pass through it. Eventually, it will pinch off completely, and then you'll have two separate bacteria. Hopefully that was a little straightforward for you. Our next type is sexual reproduction and is a bit more complicated. This involves two parents, and the offspring are going to be genetically different. Advantages of this one is that genetic variety. Again, if Lysol killed you when you did binary fission, everyone was there, but if there's that one that wasn't there, that was able to get through the Lysol, it can reproduce with other ones that can't and mix that genetic code with the other ones. This is what we're going to be calling antibiotic resistance, and that is going to be a key term. It's just not necessarily in this section. So we will go over that again. But with this genetic material and mixing of it, it does allow to cause mutations in that genetic material and therefore alterations in the genetic makeup of the different cells. Disadvantages: this is much more complex, and it's slower because you have to go through the whole process of it, plus you have to find a compatible partner. Not all bacteria are going to reproduce with every other type of bacteria out there. This is also not going to produce any new individuals. So we start with two parents and we will end with two bacteria. We do not get an increase in population through sexual reproduction, which sounds a little weird because we are reproducing, we are making different bacteria, but we are not increasing the total number of bacteria. And that will be explained in our little graphic. But this is called bacterial conjugation, sorry. Here's our process of bacterial conjugation. There we go. Basically, we do have two cells. They are bacterial. We have a donor cell and we have a recipient cell. Inside our donor cell, we're going to copy part of our genetic code, not all of it, just part of it, and that's called a plasmid. It gets there. We are then going to come next to each other. We are going to connect through a pili. We're going to send this copy of our plasmid over to the other cell. It will eventually combine into our genetic material, and you're going to get an offspring over here. I guess my face is kind of blocking it. Let's see if I can move that out of the way. I'll stick it over here. You can see over here that we do get two different cells. They do have different plasmids in it. They are both different from the original cell because our original donor made that plasmid. Our original recipient didn't have it. It then changes it. It puts it between both of them, mixes back with its genetic material, and now we've swapped genetic materials in our cells, and therefore our two cells are now genetically different from our two original ones. Let's move that back. All right.